first, let's update your biology. We used to think that genes make proteins and that's it. But Robbins makes a massive point here. Only 1.5% of our genome actually encodes for proteins. So what is the other 98.5% doing? It's not junk DNA like we used to call it. It's the regulator. It's the architect. Robbins highlights a few key players in this dark matter. Promoters and enhancers. These are the on-off switches. A mutation here doesn't change the protein structure. It changes how much of it is made. Micro-RNAs. These are the silencers. They bind to mRNA and stop translation. Long non-coding RNAs. They modulate gene expression. Why do we care? Because nearly 80% of the genetic variations found in genome-wide association studies for common diseases like type 2 diabetes or hypertension are found in these non-coding regions, not the exons. We are moving from studying protein defects to studying regulatory defects.